All biological life operates on a single, non-negotiable requirement, the continuous synthesis of adenosine triphosphate, or ATP. It is the sole molecule powering cellular function. For single-celled organisms, acquiring the raw materials for ATP is simple diffusion. But macroscopic organisms, like humans, face a massive scaling problem. Sourcing and distributing precise chemical inputs to trillions of isolated cells simultaneously is a severe logistical bottleneck. To solve this, the human body evolved three highly integrated networks: a chemical fuel extractor, a mechanical oxidizer intake, and a pressurized fluid delivery matrix. These networks operate as a physically and chemically locked sequence, forming a single serialized processing pipeline. We are going to deconstruct the architectural optimizations and the exact fluid dynamics that allow this pipeline to function. Every organ's physical shape, internal volume, and localized chemical environment exists for exactly one thing: maximizing the efficiency of ATP synthesis. The pipeline requires two primary inputs. The first is macroscopic fuel: carbohydrates, proteins, and fats. This diagram illustrates the human alimentary canal, the primary intake manifold for fuel, operating as a serialized chemical reactor. The first major stage happens in the stomach. To break down proteins, the tissue secretes hydrochloric acid and pepsin, establishing a corrosive environment. That extreme acidity is necessary for initial breakdown, but it is fatal to the surrounding tissue. If it progresses further down the tract, it destroys the internal lining. The system requires strict environmental gating. As the fluid exits the stomach and enters the small intestine, the pancreas injects a precise dose of sodium bicarbonate. This immediately neutralizes the acid and forcefully shifts the local environment to an alkaline state. That alkaline shift is a chemical trigger. It activates an entirely new set of enzymes: amylase, protease, and lipase, which execute the final breakdown of the fuel molecules. The fuel is now microscopic, but the organism faces a geometric problem. How do you physically pull enough of those dissolved nutrients out of the fluid and into the blood fast enough to keep a human alive? The solution is purely structural. The internal walls of the small intestine are heavily folded into millions of microscopic, finger-like projections called villi. These deep folds exponentially increase the surface area available to physically contact the fluid, creating a massive extraction grid within a highly confined space. The digestive tract is essentially a chemically gated reactor, precision-engineered to maximize extraction surface area. But biological fuel cannot ignite on its own. ATP production requires a second critical input: oxidizer. This schematic details the respiratory tract. Unlike the digestive reactor, this parallel intake system is driven entirely by biomechanical pressure differentials. The mechanism relies on this structure at the base, the diaphragm. It is a dome-shaped muscle anchored between the chest cavity and abdomen. When the diaphragm contracts, it forcibly flattens downward. This mechanical action actively expands the internal volume of the thoracic cavity. By expanding the cavity's volume, the internal pressure drops. This creates a negative pressure vacuum, forcing ambient air down the trachea to equalize the stace. That inhaled air rushes down an extensively branching network of bronchial tubes, splitting into finer and finer channels deep inside the chest. At the terminal ends of these branches sit the alveoli, microscopic balloon-like air sacs. This is the ultimate destination for the oxidizer. The interface required for this gas exchange is massive. If you took all the alveoli inside a single human and spread the tissue completely flat, it would cover approximately 100 square meters. Respiration is a mechanical volume pump designed to feed an impossibly large, highly optimized gas exchange interface. Once the extracted fuel and the oxidizer cross their respective barriers into the blood, the organism faces a new problem: high-speed, targeted distribution. This brings us to the mammalian heart, the central four-chambered engine driving the closed-loop delivery matrix. The internal routing is heavily segregated. Oxygen-poor blood returning on the right side must not mix with the newly oxygenated blood arriving on the left. This strict four-chamber separation enables double circulation, a specific evolutionary adaptation that runs two completely isolated pressure loops simultaneously. This diagram plots both isolated loops. Loop 1, the pulmonary circuit. The right side pumps deoxygenated blood strictly to the lungs for oxidation. Loop 2, the systemic circuit. The left side violently expels the newly oxygenated blood outward to the rest of the body. To handle the hemodynamics of this output, the arteries carrying blood away from the heart require thick muscular walls to withstand the high pressure generated by ventricular contractions. But the veins face the inverse problem. By the time blood reaches the extremities, the pressure has dissipated. Pushing fluid back up to the heart against gravity requires a different structural solution. Lacking thick walls, veins rely on internal one-way valves. Skeletal muscles physically squeeze the veins, forcing blood upward, while the valves snap shut, preventing backflow. The strict compartmentalization of the heart, paired with specific pressure gradients, ensures the continuous unidirectional delivery of materials. Bringing the entire engineering model together, we see the intake manifolds and the delivery matrix operating in parallel. The massive surface area optimizations of the gut and the lungs extract the exact raw materials required from the outside environment. High-pressure pumps and unidirectional valves force those materials through thousands of miles of vessels without stalling. All of this macro-level architecture exists entirely to serve the microscopic stakes established at the beginning, supplying the trillions of individual cells waiting at the end of the line. Inside those cells, the processed glucose and oxygen finally meet, yielding the ATP that keeps the organism alive. This integrated architecture functions as a physical resolution to the core problem of metabolic scaling. Every morphological quirk, from the alkaline secretions of the pancreas to the one-way valves in our veins, serves a single unified purpose: fueling the pipeline of life.